Now I'd like to introduce Dr. Harriet Burge. Dr. Burge is widely considered a leading expert in bioaerosols. Dr. Burge helped pioneer the field more than 30 years ago. She developed and ran the aerobiology program at the Harvard School of Public Health for 10 years and has served on 7 National Academy of Science committees on bioaerosols, published over 65 peer-reviewed journal articles, written or edited 4 books, and is a frequent speaker at conferences. Dr. Burge also served on the board of directors of the New England chapter of the Asthma and Allergy Foundation of America. And with that, Dr. Burge, I'll hand the floor over to you.
Thanks, Marvin. Good morning, everybody. We're a little tiny bit short on time today, so I'm going to pass over a few slides that are repetitious in many ways, and next time we offer this, it'll either be longer or the slides will not be there any longer. Anyway, it'll be fine. We have plenty to talk about. Some of these slides are not dealing with mold. Okay, first, the immune system. We have to talk about the immune system with respect to health effects, because virtually all of the health effects caused by the fungi deal with one way or another, deal with the immune system. Either they are actually, the symptoms are actually caused by the immune system, or the lack of a good immune system will allow the disease to occur, or perhaps the exposure damages the immune system in some ways. The immune system, as you know, was evolved to prevent infection, and we really talked about that in the last slide. I'm not going to go a lot through this. I don't know how interested you are. Now, whenever all the slides will be in the PDF, and if you have questions about anything that I skip, feel free to send a question to Dave Gallup, which the number is in your chat panel, and I will expand on what I would say normally about these. Actually, what's written here is pretty much what I would say, and I don't like to read slides at any rate, so we're not going to spend a lot of time on this. It is important to know that the immune system consists of cells able to and proteins able to distinguish your own tissues, your own cells, from foreign invaders that need to be attacked to get rid of them. And if any of that is damaged, if any of that doesn't work, then, for example, the foreign invaders will come in and cause infection, for example. Or if the immune system isn't working quite right, either the immune system itself will cause symptoms. So it's important to understand a little bit about immunology. There are two innate immune responses are there all the time, and they rely on cells that don't need training. For instance, white blood cells and natural killer cells and complement proteins. We won't describe those, but they are important as far as the innate immune system, which everybody has at birth. And so these protect you as well as they can from most of the microorganisms that are out there. There are, however, some that they don't protect against, and because the agents have special ways of getting past them, and we'll talk a little bit about that as we go along, especially with respect to the fungi. Innate responses occur really, really rapidly, and even babies have excellent innate immune responses. You have to remember that the disease organisms are with us all the time, for the most part, and at least some of them, and you have to have that kind of an immune system to survive. And it has evolved along with those potential attackers. The adaptive immune system involves two kinds of cells, T cells and B cells, and these require training not to attack our own cells. So in this case, these cells have to be exposed to some proteins on the attacking cells before they can prevent those attacks. And this is what happens when you have immunizations. You actually are injected with non-alive proteins that match those on the potential attackers. Let's say measles. You'll have a protein that matches the sites on a measles virus, and it will, and then the next time, and you will develop antibodies against that protein, and then if the measles virus comes along and you inhale one, then you won't get the measles because those cells will be able to latch on to those measles viruses and inactivate them. So it's really, those are really important. The immunizations are really, really important with respect to measles. I know there's a large measles epidemic occurring in other parts of the world than here right now because of the lack of measles vaccine. These are long-lived. The adaptive responses are long-lived. For example, with fungal allergy, if you're immunized against a particular fungal allergen, let's say Alternaria, you stay immunized for years and years and years. I'm not sensitive to Alternaria, but am to ragweed, and I was immunized against ragweed pollen, and I'm still immune to ragweed pollen as far as allergies are concerned. I'm gonna skip these because it's just a repeat. So immunoglobulins are what we call antibodies, and these are proteins that are designed to recognize. Well, they're designed to develop a mindset, so to speak, to recognize a particular substance, which is an antigen. The antigens are proteins, and almost always you can have an antigen that isn't a protein if it is attached to a reactive protein. Nickel allergies, for example, nickel attaches to an albumin molecule and makes that molecule active. So with respect to the fungi, to the molds, the antibodies learn particular protein patterns on the surfaces of the mold and attack them in that way. So there's no cross-reactivity in general between the antibodies unless well, for example, again with the mold, with Alternaria, there's several different species of Alternaria that are common, and we don't really know yet whether they cross-react, whether the antigens on the surface of those different species of Alternaria will cross-react with each other with respect to causing a protection from allergy, for example. So we don't really know that, and I'm sure somebody out there is still investigating it. I used to investigate it long years ago. So these antibodies are produced by plasma cells in response to infection. If you develop an infection, you develop antibodies. So if you've once had the measles, I'm using measles just because it's an easy one to think about. It's not a mold, obviously. You won't develop it again, at least not for many, 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 many years, and maybe never. If you're immunized against the measles, you will not develop the disease again. Again, for many, many, we don't even know how long the response lasts, but it's a long time. Or allergen exposure, and if you're exposed to allergens, particular allergens, you will stay sensitive to those for a long, long time unless you are actually treated specifically for those allergens. This is just what an antibody molecule looks like. These are the antigen binding sites right here, and they look the same here, but they're very distinctive for each. This pattern would be distinctive. And remember, you know, these are just straight lines. The antibodies are all squiggly. So go back to that just so you know. Now, there are three general types of illnesses that the molds can cause. The infection is an important one. It's not the most common one, but it's extremely important because fungal infections are often fatal, and this is partly because the antibiotics don't work as well against mold fungi as they do against bacteria and viruses. And this is because the antifungal antibiotics well, let me say this. The mold cells, individual cells of fungi, are very similar to the individual cells of people. They're all with the same structure, the same general structure. Chemically, they're different, but they have the same general structure. And because of that similarity, the antibiotics that are specific for the fungi tend to have side effects, have very serious side effects. And so it's difficult to find antibiotics that will actually attack the fungi and not devastate the person as well. So an infection occurs when a microbe enters the body and reproduces. Now, reproduces is really important. It has to be able to survive. The microbe or fungus spore has to be able to survive and multiply under the local conditions. And the local conditions meaning in the respiratory tract, for example, with temperature and pH and any kind of well, that's the first thing. Those are the local conditions. And then it has to successfully compete against microflora. Now, you know that your mouth, for example, if you open your mouth and breathe, you're going to breathe in a lot of fungal spores. And in your mouth, there are all sorts of bacteria and fungi that will compete with those invading fungi. And in general, you don't get fungal infections in your mouth that are other than the indigenous ones that live there all the time. And then they have to resist local defense mechanisms. So if you haven't been immunized, there aren't any local defense mechanisms, or not as many as you should have. And so then you can develop the disease. Once established, the organism is said to have colonized, not caused an infection necessarily, but just colonized the site. The microbe may remain dormant. Tuberculosis is a bacterium again, but it remains dormant for many, many years. It can in the lungs. And then if your immunological profile changes in some way, it can activate and begin to damage cells and cause the actual disease, the symptoms of the disease. The immune system can also cause symptoms such as fever as it tries to rid your body of the invader. All that red stuff that's around if you get bitten, scratched by a cat, which I have here, you get a redness all around, and those are the immune system coming in there to try to prevent infection from occurring, to try to attack any bacteria that have gotten into that space, onto that surface or into that cut. This one, usually when you have an infection, you get an inflammatory reaction, and that's all those white blood cells zapping, going in there trying to zap whatever it is. And then the damage occurs when you get swelling, and that inflammatory reaction increases the fragility and so forth, and then bacteria get in there and start acting, forming pus and scarring, or even death of the cells. And this happens with fungi as well as with bacteria. And we'll skip that last one. It's not having to do with fungi. So we're going to talk now specifically about mycoses, and I'll try to stay away from bacteria. It's much easier to talk about bacteria with respect to infection because much more is known. But at any rate, mycoses are infections caused by fungi. And these can be based on the site of infection, the route of acquisition, or the type of virulence exhibited by the fungus. And I think I go into that. Here's the site of infection. They can be superficial. They can be cutaneous, actually, and in the top layers of the skin, subcutaneous. Athlete's foot, for example, cutaneous, subcutaneous, systemic, which means deep infections like aspergillosis, aspergillus infections. And this is just a picture of where deep mycoses might occur on this side. All of these places, people have had these, usually with severely immunocompromised people who have seriously damaged immune systems. And then these are the superficial ones and superficial cutaneous and subcutaneous ones, like dermatophytes, and athlete's foot, ringworm, things like that. Some people classify infections on the route of entry, so they'll say airborne infections or cutaneous infections. Endogenous infection involves activation of organisms that are already present in the body, and a fungal infection that does occur from that route is Candida, where Candida is usually just a commensal and lives perfectly healthily, and both of you are healthy with that Candida there. Almost everybody has Candida somewhere on their body. But if your immune system becomes damaged, then that Candida can cause actual invasive infections. This is not common, and you have to have, it's easily diagnosed, and so this isn't the Candida that a few years ago was considered to be causing everybody symptoms of all kinds. No, this is real infection that's readily diagnosed by a physician. And then there's a classification based on virulence. This is an important one. The primary pathogens can infect anybody, normal hosts, unless they have specific antibody protection that has been induced antibody protection, or if they've had the disease before. Opportunistic pathogens cause disease in individuals with compromised host defense mechanisms. Now, this means that that some part of their immune system has been damaged for one reason or another. You can damage your immune system in your lungs, for example, by smoking. You can, if you have a transplant, for example, your immune system is highly damaged because they have to subdue the immune system so that the transplant will take and not be rejected. People with immune diseases, some cancers, HIV/AIDS, and other kinds of immune compromising diseases will allow these opportunistic pathogens to be invasive. The primary pathogens have really well-defined geographic ranges, so you're not going to come up with valley fever, for example, which is caused by Cryptococcus coccidioides in Pennsylvania, for example. You're going to have to go to the southern deserts to catch that one. Whereas in the Mississippi Valley, everybody has antibodies against histoplasma because it's endemic in the soil in the Mississippi Valley. The opportunistic fungi, on the other hand, are everywhere. Aspergillus fumigatus, which is one of the most potent of the opportunists, you can get it outdoors. Wherever you are right now, you could probably collect some of it outdoors. I'm in California, where we have it all year round. In Michigan, we had it in the springtime. There would be peaks of aspergillus outdoors in the springtime. Okay, fungal infections. All fungal infections, all require living spores for disease. And this is important because if you're going to sample and you do a spore trap, for example, you might collect a whole lot of a particular fungus and that might be dead, and you would not know that. If you're looking for a cause of an infection, you need to use culture of some sort. You can use PCR, for example, to identify what fungus is out there, but that isn't going to tell you whether those spores are alive or not. And I'm not recommending sampling at all. I'm just saying that's what you have to do. You can't. They'll be invisible if they're dead. If you use culture, if you use some kind of a spore trap, you're going to see them there, but you're not going to know whether they're the cause of what you're looking at, the disease you're looking at. Virulent agents don't grow indoors like histoplasma. I've been called several times for histoplasma outbreaks, and it has turned out that there's been, for example, a big storm that picked up contaminated soil and threw it against the walls indoors. That can happen, and then you do get an indoor exposure. But normally, these things, they don't grow. The histoplasma doesn't continue to grow in that space. If you can just leave the space, it will die. If you can't, then you have to clean it up with lots of respiratory protection because it is a virulent agent. Outdoors, it grows in specific reservoirs, and when those reservoirs are disturbed, you can have large outbreaks of some of these diseases. Not histoplasma. Yeah, histoplasma has been associated with bird colonies, grackles in particular, where people have gone in to try to remove all the grackle droppings with bulldozers and so forth, and have aerosolized lots of histoplasma, and the wind has blown it inside of schools, for example, and they've had outbreaks. You end up with outbreaks of the disease. Normally, you don't get those outbreaks. Normally, you just the disease is more subtle. It's actually quite almost subclinical in most cases. It's the immunocompromised people that develop really severe infections. These are airborne diseases, though, these virulent agents. The skin agents are probably not airborne. You get them in the gym and walking around in places where other people have them. And then, as I said, opportunistic agents are everywhere. These are the main four of the or main three. These three are the ones in this country, at least, that people are regularly exposed to. This one's restricted to the southwestern deserts, and any place else where there is actual desert environment. I've heard of some cases in Washington and Oregon. There's a big desert in Oregon. At least it's almost desert. It just has to be really dry, so there aren't a lot of other things to compete with this coccidioides. Histoplasma, we talked about, grows in soil, contaminated often with bird droppings, very wet environment, and it's very common in the east and throughout the Mississippi Valley. We don't know exactly the spread of blastomyces. It's probably similar. The ecology is probably similar to histoplasma. We don't know a whole lot about that one, and this is a South American one. So I just said all these things, and we don't need to look at that one. If you travel to an area where the fungus is common, this will raise the risk for infection. For example, if you go spend 2 weeks in the Mississippi Valley and you've never been there before, you're liable to develop antibodies. You probably won't have a noticeable infection, but you'll develop antibodies. And then this is just a list of factors that could weaken your immune system sufficient to allow these infections to become more serious. So I'm not going to read through those, but you can read them. They're commonly common in people, and one has to understand the difference between a perfectly normal person and one with any of those, diseases or conditions, if you want to predict why they have developed a particular agent, a particular disease. Histoplasma, I'm not going to do that one, and I'm not going to do that one. I have to hurry a little bit because of the lack of time today. For some reason, we're allotted less time than I expected, and so I have too many slides to cover in the hour and a half that we have here today. So remember that these slides are all in your handout, and you can ask me questions about them if you don't understand something. You're happy to answer those questions. There is a PCR assay for the detection of histoplasma. It's the only real way of deciding whether or not a particular sample, if you do try to sample the soil, because otherwise you have to inject it into mice and wait 6 weeks and culture the liver and spleen, which is ridiculous. So, okay, so whoops, that happens to me all the time. Up to 80% of people who live in the Middle West do have antibodies against histoplasma. I'm sure I do. I lived in Michigan for years. Up to 25% of HIV-infected people will develop disseminated histoplasmosis, which is a very, very serious disease. Blastomyces, we know much less about blastomyces. It was thought to be restricted to North America, but now it's been much more widely diagnosed. There's some source in nature where it comes from, but we really don't know. And this is a true endemic mycosis. It occurs mainly in immunocompetent hosts. Inhalation is the route, and it usually remains a lung infection. These things do not grow indoors ever. They do not grow indoors. Once you've inhaled it, it can be disseminated if you are immunocompromised in any way. And these are the endemic areas for coccidioidomycosis, which is a fungus that lives in dry soils because it can compete there. And it's endemic highly endemic in the southwestern deserts and in northern Mexico, and then suspected endemic in even in Washington and in a little part in Northern California, and even into Utah. But it's a desert organism. So if you're not living in the desert, you're probably safe. Unless you go visiting, you can go to Palm Springs, and if there's a sandstorm, you might be subjected to this, although it's not always there. It grows best after heavy rainfall, and I'll tell you, this year we may have coccidioidomycosis outbreaks because we have had the most rain this winter that's ever been recorded in California. It's been really, really wet. So, and then climate change is another thing we don't think a lot about with respect to the molds, but climate change will affect the distribution of the fungi in the world, and it may affect the number of valley fever infections as well as the range of coccidioides, because if a place becomes more desert-like, then the coccidioides will in fact go there, and one way or another. On the other hand, if the desert starts becoming damp and wet and starts blooming away, you won't have as many outbreaks. a flu-like illness. Most people who breathe in don't get sick at all, and people who do get sick will get better on their own without treatment. You can get antifungal infections. Immunocompromised people should do that. It's very contagious when you grow it in culture. So labs, environmental microbiology labs, don't like to grow coccidioides in culture. It's very, has to be very tightly controlled. And they rarely have to, because we really know how people get this disease, and so we don't worry too much about it. Now, the opportunistic infections are the ones that we really do need to worry about, because these organisms are present everywhere. And there are serious, serious issues for immunocompromised people with AIDS and organ transplants, chemotherapy, bone marrow transplants. Normal people almost never develop invasive fungal infections. Our own immune system, without any antibodies, is able to fight these organisms off. They're not that strong. Aspergillus fumigatus is the one that, Aspergillus fumigatus is the one that is most likely to be able to get through a minimally compromised person, because it has toxins that actually help get it past the immune system. But I've been working with Aspergillus fumigatus for my whole career, and I certainly have never been infected, and normal people really are not infected. These are other opportunistic mycoses. Candida albicans is opportunistic. As I said earlier, if you become immunosuppressed, you can develop infections, actual infections, but mostly it's just a symbiotic thing that's growing with your other microbes that occupy your body. Several species of Aspergillus. The zygomycetes, these are a particular class of fungi, and there are several of them that can cause human infections. There was one outbreak where a helicopter landing on top of a hospital was contaminated with, there was a pond up there, not a pond, but water had accumulated, and rhizopus had colonized that water, and the helicopters were forcing the water through the ventilation system into the hospital, and there was an outbreak of rhizopus infections. Now, that's uncommon, but it's something to think about when you're trying to figure out why there's an outbreak. We talked about this one and this one. Candidiasis or candidemia are both the same thing. Intensive care units in hospitals, that means you're really, really, really sick, and your immune system is probably compromised to some extent at recent surgery. If you have a central catheter, a central line catheter, those catheters sometimes become contaminated and introduce this organism directly into your bloodstream. And so that's a potential danger that people watch for. And there's no evidence that Candida causes all the physical and psychological problems that people have complained about. And this is more or less died down, so that's a good thing. Okay, Aspergillus is a true opportunistic infection. Major concern, it's often fatal. The number of living spores required to initiate infection is unknown, and so when you sample, if you try to sample for this, you don't know if it's possible that one single spore could cause an infection in a suitably compromised person. On the other hand, farmers deal with inhale millions of these spores every day when they're dealing with hay that has become moldy, and they aren't affected at all, or at least not with infections. So it's compromising, immunocompromising is the main risk factor. There are actually, aspergillosis comes in 3 different forms. Invasive aspergillosis is actually the infection that we've been talking about. Aspergilloma occurs when holes in the lung parenchyma, for example, if you've had tuberculosis and you have empty spaces in your lungs, Aspergillus can colonize those spaces and form fungus balls. It isn't really an infection. The Aspergillus just sits there and releases allergens, of course, but it normally just sits there. And then allergic bronchopulmonary aspergillosis. This happens in the lungs of asthmatic people who have lots of mucus in their lungs. And the aspergillus colonizes that mucus in the lungs and causes severe hypersensitivity reactions. It's not an infection. It's the colonization of that fungus, and the allergens are just continually released into the system of that person. It's not generally treated with antifungal agents. It's treated with steroids to get rid of the mucus, because that's what needs to be. As soon as the mucus is gone, the aspergillus will go away too. Obviously, that's a gross oversimplification. In some cases, you do have to attack the aspergillus and try to get rid of that as well. But as I said, fungal antibiotics can sometimes have bad side effects, well, as does the prednisone that you might use to reduce the mucus. But this is a complicated disease to treat, and very, excuse me, very unpleasant. These are just some of the kinds of zygomycetes that might cause infection. Mucor, rhizopus, and those mucor and rhizopus are the most common that cause infections. And these are the ones that have just been reported. There are probably others, we don't know. These are inhalation diseases, all of them, and percutaneous. For example, if you stuck yourself with a needle that had this particular fungus on it, you could develop a local infection. And apparently you can ingest the spores and have an infection. I haven't read too much about that. There's a list here of somewhat different immunological damage that that allows these kinds of infections. Less serious than some of the ones. I mean, well, diabetes is not serious, but it is very serious, in fact. But it usually doesn't lead to fungal infections, except with these rhizopus-related things. Chronic prednisone use, meaning that you've damaged your immune system over time, and so forth. There's a whole list of things that could damage your immune system, and this could be at least a partial reference list for you to look at those kinds of things if you are working in that field. Cryptococcus is it used to be considered one of the virulent infections. It's really not. It's really most of the species are opportunistic. It's widely distributed and relatively common in immunocompromised hosts, even slightly immunocompromised. It's interesting that Cryptococcus has been found in the ruins of Chernobyl, and it may be able to use radiation as an energy source. That would be a scary thought. There's one species, I don't think I have that in here. There's one species, Cryptococcus gattii. Yeah, here it is. That is a tropical species, and it's been reported in the northeastern North America. Actually, that should be northwestern North America. That should be northwestern. I'll have to correct that. Because Oregon, there have been 60 cases were identified in the U.S., Oregon and Washington, and one each from Idaho and California. These were only 50% of them were immunocompromised. The other 48% were not. And so this is one of the cryptococci that can actually infect normal people. And it's probably a matter of dose. The more you get, the more of a particular microorganism that you inhale or get into your system, the more likely you are to develop an infection. These are skin infections. We're not going to go a great deal into these. They don't have really an environmental, they have an environmental source, but it's not airborne. It's certainly not. Carpet, these things last in carpeting, for example, with skin. They live on the skin scales in the carpeting, and that's where you catch them in the gym, in gymnasia, where the locker rooms are damp. Is a really good place to develop dermatophytes. But there's no evidence that that they are airborne, that they're transmitted by the airborne route. They actually live in the keratinized, the surface layers of your skin, the keratin is a protein that's on the surface. And these are just some of the names of the symptoms. You name this by symptom. If it's a little round thing in your hair, you call it ringworm and so forth. I skipped these. Whoops. What happened?
We can still see your screen. If you just want to pull up the... Okay.
What happened?
If you just want to pull up your PowerPoint.
I lost it.
We can still... Yeah.
Okay. That's weird. Okay.
Okay,
there we go. Okay, hypersensitivity. Hypersensitivity is the most common ailment that the fungi cause, and the fungal allergens can cause asthma and hay fever and allergic fungal sinusitis. In other words, the stuff grows in your fungi and the allergens are released into your system, and allergic bronchopulmonary aspergillosis. That's a hypersensitivity disease. Let me get back into full mode here. Here we go. Hypersensitivity reactions are classified by mechanism. The kind that I just talked about, ABPA, allergic bronchopulmonary aspergillosis, asthma, hay fever, are all involve IgE, immunoglobulin E molecules, and you are exposed to, if you have the type of genetic pattern that allows you to produce excess IgE antibody, then the antibodies are formed and learn to recognize a particular antigen, like Alternaria. I use Alternaria a lot because it's the most common allergen. Alternaria and Aspergillus fumigatus are the two most common fungal allergens that we see when we skin test people. And then that's the immediate hypersensitivity diseases like asthma. You're exposed, you develop the symptoms, and then you get better. I mean, you get treated and so forth. And hay fever the same way. There's another kind of delayed hypersensitivity that you get that you can get with exposure to fungal allergens. And this is a cell-mediated response. It's a little more complex than that, but for now we'll call it a cell-mediated response, where it takes exposure over quite a long period of time before you, and massive exposures over a long period of time, relatively speaking. Now it can be weeks or it can be much longer than that, and it's a different kind of disease. So this is the process of, really quickly I'll go through this, a process of developing allergy, type 1 hypersensitivity. It has ragweed. You can pretend this is a fungal spore. It's the same response. It attaches to a B cell. Here's the B cell. And the B cell turns into a plasma cell. Don't ask me how that happens. I'm not a physiologist. But then this plasma cell produces all these IgE molecules, and they're all sensitive. They're all specific for ragweed pollen, all of them, all these. And it takes a whole bunch of exposure. It takes time after time of exposure. When I moved to Michigan from California, I'd never encountered ragweed. It took me 1.5 years before I started being sensitized to ragweed, and then it was all downhill from there. So it takes a lot of relatively low-level exposure to develop these antibodies. And then if they attach themselves to these mast cells, and as long as they're just sitting there, it's okay. But then if you're exposed to another ragweed cell, I mean, this is after your weeks or years of exposure, then if it's another cell that's specific for these attaches, then this mast cell breaks open and all these chemicals are released from these little vacuoles in here, and you develop symptoms. This is histamine, for example, is one of the chemicals that you develop. And so if you use antihistamines, you're blocking right here so that you don't get the symptoms. So this is more just a description of what those cells actually are. I'll leave it here for you to look at. If you have questions about it, by all means ask. This is important because if you have intravenous, if you actually have an intravenous injection of an allergen, you get anaphylaxis. Now, usually we don't do that with mold allergens. Radio contrast media are what happens if people have a number of episodes of radio contrast injections. They can develop severe allergy, anaphylaxis. This is a very, very dangerous condition where your blood pressure falls. I hopefully have a slide that talks about this, because right this minute it's not coming into my head. I'll come back to that. Intradermal is the local release of histamine. It's interesting because I apparently have cat allergy, and my cat scratched me the other day, and my arm swelled up, and that was a local release of histamine related to that cat allergen that got into the surface of my skin. Not going to get rid of the cats, however. Inhalation, you get allergic rhinitis. In the upper airways, you get allergic rhinitis. Hay fever, that's what this is, hay fever. In the lower airways, you get asthma, local release of histamine right in your lungs, and you develop asthma. If you actually ingest an allergen, you get local release of histamine and diarrhea and vomiting. And actually, food allergy is one of the most common triggers for anaphylaxis, which starts with diarrhea and vomiting. That's one of the first symptoms these people experience who do this, and passing out is the next one. I have a friend who ends up with her face in her spaghetti if she happens to get any egg in her food, and very dangerous disease. Diffusion into the bloodstream, that's what happens. So you local release of histamine, and then diffusion into the bloodstream, you get hives, and then anaphylaxis. And some people are so severely sensitive to these that even a tiny bit of whatever it is, whatever you're in, peanut allergy or any kind of an allergy like that that's by ingestion will develop the anaphylactic symptoms instantly, and if they aren't taken to the hospital immediately, or at least injected with epinephrine immediately, they will not survive. Ah, that was nice. That's a way to get quickly through. Okay, we did that one. Exposure to fungal allergens is very high, and sensitization is common, at least 10%, probably more, because the only way we know whether somebody is sensitive to an allergen, a fungal allergen, is if they are actually skin tested, and we have the proper skin test material to test them. And that often does not happen because there's only a relatively small proportion of the fungi that are out there have been used for skin testing. So it's probable that this number is much higher. The most common ones that we see are Alternaria, and it's interesting because Alternaria is not the highest concentration in the outside air. It just has very active antigens on its surface. Low doses over a long period of time are needed for sensitization, and then once you're sensitized, you need higher doses for symptoms. That sounds weird, but it's the case. And then fungal sensitivity. If you are allergic to the fungi, you're more likely to have asthma than someone who's not allergic to the fungi, who just is allergic to ragweed, for example. Most fungal exposure occurs outdoors. People do worry that they've got mold in their houses and their allergies are because of the mold in their houses. It's difficult to tease that out because if they're walking around in the summertime in Pennsylvania, for example, they're being exposed to this, what you see right here. Actually, this is Michigan, but same difference. These are all these little things here are fungal spores, Cladosporium, Alternaria. There's little basidiospores in here. There's all kinds of stuff in here. This is a pollen grade, by the way. Pieces of fungi, little pieces probably of Alternaria. So you're exposed to that all the time. And then the fungus has to actually, many of the fungi have to actually germinate. Now, Alternaria is an exception to that. It can it can be released immediately, but cladosporium, for example, the spores actually have to germinate on your mucous membranes before the reaction occurs. They don't go on to grow. They die once they germinate because they don't have the proper environment. But they release enough stuff after that germination. So you might be exposed outdoors to masses of stuff, and then after you get indoors a couple of hours later, you develop the symptoms, and then you think, oh, well, I must have gotten these in my house. Not necessarily. You could easily have been exposed outdoors, walk into your house, had a cup of coffee or a drink or whatever it is you do, lay down, and then develop the symptoms, or your office or any place else. So diagnosing where an exposure, detecting where an exposure to fungal allergens occur is not straightforward. You need a fungal strategy seminar in order to explain all that because it's really quite complex. This is cladosporium growing on a wall, and this is the kind of thing you see in houses where people think that the mold in their house is causing symptoms. It could certainly be, although cladosporium doesn't release a lot of spores into the air from this kind of growth. It needs active air movement to get the cladosporium off the walls. So this could be just absolutely innocuous, sitting there, not hurting anybody except looking bad and smelling bad. On the other hand, it could in fact be causing allergies in some people. The frequency of cladosporium positive skin tests is relatively low, but that could be due to the lack of good skin testing materials, or it could be that cladosporium isn't very allergenic, doesn't have good antigens. That was for the immediate type allergy. These are fungal colonization diseases. There are two: allergic bronchopulmonary mycosis. Now we call it mycosis because it can be several different fungi, not just Aspergillus. Aspergillus is by far the most common, and it's almost always Aspergillus fumigatus. Allergic fungal sinusitis is another one where the fungi grow in your sinuses. Now that one does not necessarily, Aspergillus isn't necessarily the most common one there. In the southeast, some of the dark fungi, dark spored fungi like Drexler, bipolaris, those kinds of things, curvularia, those kinds of fungi may cause fungal sinusitis. And in this case, the fungus grows in the mucus in your sinuses and produces masses of mucus and masses of protein and masses of cheesy stuff. It's very uncomfortable and treated generally with surgery. The sinus is filled with fungal hyphae, actually, and thick mucus. The risk depends on having chronic asthma or chronic rhinitis. People without asthma or rhinitis do not develop these diseases. The food for the fungus is the mucus that's produced, the massive amounts of mucus that's produced by these diseases. And then only a few fungi are involved, as I said. A little bit for asthma. Extensive evidence documents that exposure to fungi precipitates asthma attacks once the initial sensitization has occurred. And fungal exposure may, in fact, trigger asthma in the first place. It may be the first. See, once you have asthma, then other things can join in the party, and you can become sensitized to a number of things. The first precipitating factor is often house dust mites or cockroaches or something like that, but it has been shown that fungi can do that as well. I was on a committee where there was long arguments about initiation of asthma or exacerbation of asthma, so that's where that comes from. That one is just one, and you have this in your thing, you can read it. It's just one case that I thought was very interesting because the asthma attack was related to fungi growing on a mouth guard, and it wasn't due to the school at all. It was due to that moldy mouth guard. And they thought the school was contaminated, and it really wasn't, because the kid only had asthma at school, never at home. Didn't wear the mouth guard at home. 3 and 4 hypersensitivity. This is a delayed hypersensitivity, and this is hypersensitivity pneumonitis, which is also called allergic alveolitis, especially in Europe. Exposure to the antigens of these organisms, Aspergillus is one of the organisms. Penicillium does it. There's a number of different, some mushroom spores. Exposure to them leads to exposure to these antigens leads to antibody IgG molecules. These are called precipitating antibodies. And then re-exposure to these antigens. If you continually re-exposed, you get antigen-antibody complexes that are quite large and develop in the alveoli, and eventually cause fibrosis, pulmonary fibrosis. These are just examples. These are just more how you if you want to know all these the actual chemicals involved and so forth. This is just a brief summary of some of them. And if you want to know more about this, send me a question. I can send you some references to help you with this. Otherwise, at least you understand that it's not just a straightforward thing that, that as straightforward as it sounds. I think immunology is very complex and difficult, but maybe some of you are immunologists and don't think that. There's two different kinds. The subacute kind, where you get interstitial fibrosis, bronchitis with or without pneumonia, and this particular stage can be reduced or reversed by eliminating exposures. So most of this will go away. Interstitial fibrosis does not go away, but if you catch it soon enough, this will be relatively minimal, and you can survive with that. This is the chronic forms. You get this is where the alveolar destruction occurs, and you get interstitial inflammation and fibrosis, and continuing exposure can and has led to death in many cases. Acute symptoms. Oh, this is, by the way, of somebody playing with moldy hay. He's not playing, but he's working with it. It's a hay bale that was out in the field, not protected, and it developed mold. Now, this is probably not a mold, actually. This is probably a bacterium called an actinomycete, which is a filamentous bacterium that makes spores. But there's also mold in this mixture. And these spores are notorious for causing hypersensitivity disease. And anybody with any industrial hygiene training would certainly not do this without good respiratory protection. These are 1 micron spores, so you have to have, they are particles, but you have to have respiratory protection if you're going to do this. And these are some of the symptoms that you get. Intense exposure to small particle organic dust, including small fungal spores. Now, this is how hypersensitivity pneumonitis actually starts. We call it HP. Actually starts. And then if you get rid of the organic dust, get rid of all this exposure, put a respirator on this guy who's out there in the field doing these things, the symptoms will go away, essentially, and you'll be better, and especially if you do it right away as soon as these symptoms start appearing. If you don't get rid of this, if you continue to be, you'll be sensitized. You'll be sensitized at this point. And at this point, very low level exposure over a long period will continue to produce the same symptoms and getting worse and worse and worse. So it's really important to recognize early stage hypersensitivity pneumonitis so that you don't come into the chronic stage. A lot of things cause hypersensitivity pneumonitis, and they're called by the name of the, like malt workers' lung. These are malt workers, moldy barley, peat moss. These all the way down here, tree cutter lung, penicillium, dry rot, Marulius. This is one of those wood rotting fungi. It goes on and on. I mean, this is just some. I could have more. I could show you more. Endless ones that have been described that causing hypersensitivity. Endless fungal causes. There are actually a whole lot of other causes as well, but these are only fungal causes of hypersensitivity pneumonitis. Let me check the time. We're doing really good. Okay. Just half a second. My voice gets tired after a while. Now, fungi do produce toxicoses, and this has been the bane of my existence over the last 30 years is the exaggerations of the potential toxicoses that can happen. But in fact, fungi do produce toxicoses, and we're going to explore that for a bit. Toxic fungal metabolites. If they're toxic mainly to bacteria, they're called antibiotics. If they're toxic mainly to plants, they're called phytotoxins. If they're toxic to the animal kingdom and are produced by macrofungi or mushrooms, they're called mushroom toxins. And if they're toxic to the animal kingdom and produced by microfungi, meaning like Aspergillus or Stachybotrys or any of those little microfungi, they're called mycotoxins. So mycotoxins are the ones that everybody seems to be concerned mostly about. Mushroom toxins kill a lot of people every year who go out and collect mushrooms and then eat them, not knowing really what they are. Lots and lots of fungi are plant pathogens, and those plant pathogens produce these phytotoxins as part of their infection process. And then we know about antibiotics. Okay, again, classification schemes tend to reflect the training. For example, clinicians call mycotoxin. Now, these are the real mycotoxins produced by microfungi, produced by Aspergillus and Stachybotrys and Penicillium and Fusarium and all these microfungi that that everybody seems to be worried about these days, but that have always been around. They have always been there. Anyway, the organ affected. The clinicians call them like hepatotoxins for liver, nephrotoxins for kidneys, immunotoxins for the immune system. And there are there is evidence for all of those things happening with specific mycotoxins under some circumstances. The cell biologists call it by mechanism of action, teratogens, birth defects, mutagens, carcinogens, allergens, and then organic chemists by the chemical structure, lactones, coumarins, and so forth. So it's very confusing when you read the literature. I came up with this slide because I was reading lots and lots of mycotoxin literature, and every one of them called them different things. Drives you crazy when you're trying to make a reasonably concise presentation. Acute versus chronic toxicosis. Acute toxicity, a single exposure or multiple exposures. Chronic toxicity, prolonged, repeated exposures, or internal exposures because the substance isn't metabolized. And the doses required for acute toxicity are generally much higher than those for chronic toxicity. That seems a no-brainer to me. Is a disease actually caused by mycotoxin exposure? Well, mycotoxins do cause disease. They cause disease in countries where people are forced to eat moldy food. Ochratoxin is well known in some countries where ochratoxin contaminates grains and even all kinds of foodstuffs, and they eat that food and develop the symptoms of mycotoxin exposure. So it certainly has caused symptoms. It has caused symptoms by ingestion. There's anecdotal evidence only that mycotoxins have caused symptoms by the airborne route. There are animal studies that document that effects can occur in animals, with airborne exposure, but that doesn't really, I'm sorry, not with airborne exposure, with injection. So we have all this evidence that, of course, mycotoxins cause damage if you're actually exposed to them. If you're actually exposed to a sufficient concentration of a mycotoxin, yeah, you will get sick. You will have problems. These are ways that there have been evidence, anecdotal evidence, animal studies, epidemiology, and biological monitoring. Those are 4 different ways that you can document whether or not a mycotoxin is causing problems. So we're going to, for the moment, other than the fact that we know that mycotoxins cause disease if the exposure is appropriate and concentrated enough, what we don't know is whether inhaling spores that make mycotoxins will cause these same diseases. Now, in my opinion, if you inhale enough of those spores, of course, it will cause symptoms. The question is, how much of that do you have to inhale and over what length of time? For example, if you do inhale spores, how long are they going to last in your system? Are they going to just stay there forever? Probably not. Are they going to die? Are they going to be attacked by immune systems and white cells and so forth? Yes, they are. So they're going to be present for only a reasonably short length of time. So you're going to be exposed to the toxins during that time. Are the toxins metabolized? Probably. Yes, they are. So they're going to gradually disappear too. So all those factors need to be taken into account when you're trying to decide whether or not a particular moldy household is causing toxic symptoms in anybody. A few anecdotal reports in the peer-reviewed literature indicate the possibility of an association between airborne mycotoxin exposure and illness. Most of the literature says that the association is not documented. Admit that the association is not documented. It's just an anecdotal association. Unfortunately, the courts accept that as evidence, and the courts only need 50-50 chance, so that's the courts for you. There are many reports of animal studies of mycotoxin effects that talk about the mechanisms of action. And when you do an animal study, you don't want to wait 2 years before anything happens. So they do high doses, much higher than you would ever expect from anything other than ingestion. Now remember, we know ingestion works, but we don't know about this environmental exposure, airborne exposures. Many of these studies are well done, and they indicate that mycotoxins are in fact toxic, but we already know that. We already know that. And these are seeking to tell why they're toxic and what they actually do to cells with high exposure. They don't establish a connection between exposure and disease. I apologize for spending so much time on this, but I'd say 80% of the questions I get from not from these seminars, but outside questions that come in have to do with odd diseases that are being blamed on mycotoxins. And I always have to write back and say, you know, there's no evidence that this ever occurs. Several epi studies have associated chronic mycotoxin exposure with cancer. Now, these are all occupational. This is peanut handlers, people who handle peanuts regularly in the Netherlands have a very, very slight increase in nasopharyngeal cancer due to aflatoxin exposure. Small risk, and yet they're exposed to aflatoxin all the time through the air. There's one epidemiological study that reported Stachybotrys causing pulmonary bleeding in infants, and this was improperly done, and the CDC published an analysis saying that the conclusions were not justified. So this was the only real evidence of airborne mycotoxin causing a specific disease, and the CDC says that the conclusions were not justified. So we can't depend on this. We have to say, you know, okay, fine. We still don't know. We still don't know whether it works. Biological monitoring is really a good way of doing these kinds of studies, but most of the studies on biological monitoring are from food exposure. In food exposure in the countries where a lot of food exposure to mycotoxins occurs, and there's really good evidence that these work and that people are being exposed to mycotoxins, and probably that there are symptoms related to that. But we already know that food works. A few studies claim detection of mycotoxins in bodily fluids of people reporting residential exposure. So far, in my opinion only, these studies are not convincing, and this is primarily because of bias. The people doing the studies have a financial interest in there being a connection, and not purposely. Nobody does bias. Nobody is purposefully biased. But it's very hard to eliminate bias. Scientists spend endless amounts of effort trying to document that they aren't being biased. And I know I did it myself. You're trying to make sure, okay, am I trying to prove this, or am I trying to find out what's really going on? That's exactly the problem. So, so all this adds up to the fact that right now, I don't think that there's any good case for airborne mycotoxin exposure in moldy homes to routinely cause illness. Now, there may be exceptional cases where it does, but the first symptom of a heavy Stachybotrys exposure, for example, would be rashes on your mouth and your nose and your mucous membranes. And I've never seen that. Nobody has ever complained of that. They start complaining of neurological symptoms and chronic fatigue symptoms and things of that sort. And that's not going to happen in that way. You're going to end up first having the normal symptoms that even people who eat mycotoxins, the first thing they get is mucous membrane symptoms. So lower respiratory illness. Now I'm not talking about fungal infections. I'm talking about bronchitis, for example, from a bacterium. Does exposure to mycotoxins increase the susceptibility? Does it damage your immune system so that you have an increased susceptibility to lower respiratory infections? The data for this is based on very large epidemiological studies in children, and there's a small association between having damp home interiors and increased lower respiratory illness in young children. Whether or not that's caused by fungi is unknown, absolutely unknown even today, and it seems a bit doubtful, actually. So we'll have to keep monitoring that literature. This is Stachybotrys. This is a picture of it. This is it growing in my lab manager's house. No symptoms whatsoever. It was growing in his basement, and he just ignored it. We're not going to go through all these. We're going to talk about irritants, because this is another possibility that people ask me. If a fungus is growing in an interior space, it is releasing volatile organic compounds that are responsible for the odor, the moldy fungal odor. There is not good evidence yet that those volatile organic compounds, gases released by the fungi, are actually responsible for any kind of illness. They're unpleasant, and so there's a psychological problem associated with them, but not any direct. In fact, there's evidence that there is no effect, because they're odorous in very, very minute quantities. So you're being exposed to very tiny, tiny amounts. And right this minute, as far as I can tell from surveying the literature, there's no real good evidence that those cause any actual physical effects. They do cause psychological effects. I mean, I don't like living in moldy houses either, but yeah. Okay, we've done, I'm going to check my time here. We've done most of this, and I don't want to get too much into this, because I think that the toxins, yeah, here's just a summary. Hundreds of publications of dietary mycotoxins in underdeveloped countries. No publications that provide evidence for airborne mycotoxin exposure sufficient to cause illness. Sufficient to cause illness. And this is just an aside. Stachybotrys can be growing all over your walls, but the conidia don't get off the walls, because the air speeds have to be much higher than they normally are inside. And you can read the internet. I have a nephew living with me who learns everything he knows off the internet, and we get into arguments all the time, because people claim anything on it. You could write anything you want there, and none of it's peer-reviewed. So they say that scientists, including me, I get attacked by this all the time, underreporting dangers of toxic mold, and that is just not true. We've investigated and read and messed around it. It just doesn't. Position papers by major medical organizations are considered biased. Well, you know, the people who are writing on the web are the biased ones. These people take great effort to not be biased, and why should they? Why should they say that there's no effect when there really is? It would be ridiculous for people of this sort to do that. And expert witnesses are considered biased because they're paid for presenting their opinions. Well, I'm an expert witness a lot, and I've had the opinions for my whole career, like 40 years. Just because somebody's paying for them doesn't mean I've changed them. for years and years and years, I presented those opinions with no money at all. So consider that with expert witnesses. Now, there are expert witnesses who are biased, and I probably am biased to some extent. Maybe I'm biased against mycotoxin exposure. I don't know. I try to avoid that, but anyway. Okay, you can read that. These are references for some. These are older ones. I'll have to update this and give you some better ones, some newer ones. And these are general references that that cover a lot of what I've talked about. Today that might help you. More general references and sites, websites that are interesting and have lots of information about mold and immunobiology and so forth. Immunobiology is really interesting if you can have the time to explore that. So we have a few more minutes. Actually, I think we have 15 minutes. Is that right, Marvin?
Yeah, according to my clock, yeah, 15 more minutes. So did you want to
I sped through this, so I think this is be more interesting to just go on and have people ask a few questions, because I'd really like to know what where the interests lie. Anyway, I'm happy to take questions.
Okay, great. And just as a quick reminder to our audience members, if you have a question, please enter it into the questions and chat panel located within the lower right-hand corner of your control panel. So we'll give it some time to see if anyone has any questions. In the meantime, we do have a question in the queue. Do you have a high aerosolized spore count to have mycotoxins exposure?
You have to have a very, very high spore count. And we calculated it, one of my doctoral students and I calculated it years and years ago and developed a model about how much exposure you'd actually have to have. And it was 8,000 spores per cubic meter consistently over an 8-hour exposure period. She's an industrial hygienist, and she liked that over that form of doing it. But that's a lot. You never see more than 200 or 300 in an airborne state, and then they're transient, and usually they're there because somebody has disturbed the colonies, not because they're just there all the time. If you go into a quiet room with no disturbance, with Stachybotrys on the walls, you're not going to get a lot of. You might get 1 or 2 spores, but that's going to be it. And so when you see 1 or 2 spores on a spore trap sample, that means there might be mycotoxin, there might be Stachybotrys, for example, growing somewhere in the space, but that doesn't mean that you've got a disease-causing exposure at all, not at all.
Okay, great. Next question. How do you detect and measure mycotoxins within a building?
You don't generally. There are what people generally look for is a mycotoxic fungi, and or you can take the mycotoxins, you can take the material the mycotoxin is growing on and try to analyze it. The problem with mycotoxin, it's a chemical analysis, and you need a lot of material in order to get the detection limit, to get up above the detection limit. There are people who claim to have developed immunoassays for some of the mycotoxins. I haven't really liked those so far. And again, some of them have been developed, many of them have been developed by people with serious biases toward mycotoxin exposure being important. So the question is not answerable by me at this moment. I would be happy if you'd like to leave the question in the queue, or we can send it to Dave and ask. I can look into it a little bit more to see if anything just in the last couple of years has come up. I really am retired, more or less, and so I don't read all of the literature all of the time. But during these seminars, I do read a lot, so I'm happy to look that up and give you a better answer.
Great. And that same person asked kind of a similar question, how do I measure and detect MVOCs within a building? that different?
MVOCs, you can use the EPA methods, the evacuated canister method. That's done commonly. I haven't. That one is the best. I think, in my opinion, it's the best if you know how to do that. And then it has to be. It's a GCMS gas chromatography mass spectrometry method. It's a chemical method, and that is used quite commonly to detect MVOCs, and that detects specific ones. It'll give you a range of, it'll give you aldehydes and ketones and so forth. Or you can use a mold dog. I have a friend who uses a mold dog. She has a whole little batch of them, and they work. She has 100% so far. When one of her dogs has sniffed at a wall and barked, she's opened the wall and found mold 100% of the time. Now she is biased. There's no doubt about that, but people poo-poo that a lot. I know my dog can. We have 2 dogs, and if they can find food anywhere it is, I mean, it doesn't matter. It can be outside under the fireplace, and they'll find it. They know how to find things with their noses. That's what they're good at. So anyway, that's another silly. It's not silly, actually. Hers, it works very well. She's a Canadian woman. Anyway.
Okay. Next question. Well, someone's asking, how do you collect a sample for it to be examined? I believe maybe they're talking about black mold spore.
Okay, if you want the sampling, we have a whole seminar on sampling, but just for reference, if you have what looks like mold growth and you want to know what the name of the mold is, which you might, you do a tape sample. You just do a cellophane tape and put it on a slide. There actually are, you can buy little things that do this that are already sticky, but you can use cellophane tape. I've used it for years, and put it on a glass slide, or at least on something where the spores will stay stuck to it. And a glass slide is nice because then we don't have to remove the tape in order to read it. And then it's looked at microscopically, and we can tell you the name of the fungus. That's a really nice way of doing it. If you want to know what's becoming airborne, you have to use air sampling. You can't just guess. You have to use air sampling. So in that case, if it's a fungus that, well, there's 2 things you can do. One is if you have fungal growth, you do tape 1 or more tape samples and have those analyzed, and then you do air samples, and then you can compare what's in the air. We're using a spore trap, a little spore trap. And I'm not, you can contact EMLab, which I recommend, and that one of the project managers will talk to you about exactly how to do this. They're really well trained. But then you can compare what's in the air with what's on the wall, and it'll tell you whether or not what's on the wall is getting into the air. A key thing to remember is to test the air before you start messing with the wall, because if you start taking tape samples off of a wall, you're going to disturb the spores in a way that they're not normally disturbed. And what you want to know is it getting in the air when it's not being disturbed.
Okay, great. Next question. What levels of mold exposure measured before you'd want to close a building?
Well, it depends on, there's a million reasons for closing a building. I worked with a building in Hawaii where it was closed because the owners didn't want to take the risk of people becoming sick because there was mold in every single room. And it was really obvious mold, and so they closed it, whether or not it would have hurt anybody. If it's a matter of the public being in and out of those buildings, I think if you see a lot of visible mold growth, they're going to have to, I mean, in this case, it was growing on surfaces all over the place. Then you're going to have to evacuate the building, obviously. I think it's more than saying how many spores are in the air. I think it's more than saying how extensive is the mold and where is it. If it's mostly just around the baseboards and it's wet mold, you probably can just, seal off those areas and remediate without evacuating an entire building. If you disturb powdery mold on surfaces, you're going to put it into the air. So if you're going to do remediation of powdery mold, you're going to have to either do it in really good containment, particle containment, or you're going to have to evacuate the building and have the workers wear respiratory protection. So that's more what I look at rather than how many spores there are. It's how much exposure is going to be caused by remediation. The remediation exposure is going to be much higher than any exposure that people get just by walking in. If people are afraid to go in the buildings, I actually have been in buildings where there was mold, massive amounts of mold, even in the air, and the buildings were not evacuated. There were people still living in the buildings. They complained a lot, but they were staying in those buildings. And even during remediation, I don't think they had them leave. It was a government building, so who knows. So, and nobody was getting direly ill, so I don't worry too much about that. But during remediation, yes, I think either evacuation or really good containment.
Okay, great. Next question. Someone was wondering if you could please discuss moldy odors. Is there a mold problem if the odor is present?
There certainly are molds somewhere. Now, I'm sitting in my study here, and I have to keep the door closed because one of my dogs will chew everything in here. And when I open the door, I can smell mold, a slight amount of mold, and I'm sure that there is some somewhere. It's probably in the closet, coming from the closet. So yes, if you smell mold, if you smell that moldy odor, you've got mold. Is it serious? Not necessarily. You know, with my closet, if I would just keep the door open and the light turned on for a while, it would go away. If you have a moldy basement, if you put a good dehumidifier, not just one of those that you have to go down and empty all the time, but a really good commercial dehumidifier that you can drain into a wash basin or something, the mold will go away. The mold smell will go away, and the mold will die. So yes, it does indicate mold, as long as you know what a moldy odor is. I mean, I assume most people do.
Okay, next question. Is there a movement away from Anderson N6 culture sampling for the alternative methods?
No, as far as I know, not. I hope not. The Anderson sampler, well, we use the biocassette, which is an Anderson N6 surrogate, shall we say, that works really quite well. If you're going to look for living microorganisms, you have to use culture. And the Anderson is the most efficient sampler. The original Anderson sampler is probably the most efficient sampler out there as far as collecting the small spores that you're worried about, like Aspergillus fumigatus. I'm going to present a thing at the AIHCE about that, about sampling in hospitals where you're trying to trace infection sources, infectious disease sources. So yeah, I think that the majority of samples that we get are not culture. They're mostly spore traps, and this is because people aren't looking necessarily for the causes of health effects. They're looking for whether or not a space is moldy. And those are two really different questions. If you're interested in that, our fungal strategies seminar is a really good one for that. That's where I focus a lot on what kind of strategy you use and under which situation.
Okay, next question. What effects can mold exposure have on someone being treated for chronic Lyme disease?
Well, it depends on how you're being treated. In general, I would say not much, because you're being exposed to fungi every time, masses of fungi every time you step out the door, and you're breathing in those fungi. And as far as I know, they don't have any additional effect unless you have severe allergies, mold allergies, on top of the Lyme disease. It depends on how you're being treated, what you're being treated with for the Lyme disease. If you're being treated with something that damages your immune system, which I would doubt, but maybe that is a case. I'm not really very familiar with Lyme disease. But if it damages your immune system, whatever treatment you're undergoing, then I would try to avoid molds growing on surfaces, large quantities of molds. I would try to avoid that. household mold, for example. If you have, you don't want to live in a moldy house, but you're never going to be able to avoid fungi completely. It's impossible. And that's the problem with people who have severe immunodepression. It's almost impossible to avoid exposure. And a great effort is made to prevent exposure. And even just forgetting for a little bit that you have to prevent exposure can be deadly. So yeah, I haven't heard of any association between fungi and Lyme disease, none whatsoever.
Okay. Looks like we have time for maybe one more question here. The question is, do you recommend running a negative air machine before testing a room for spores?
No. Well, it depends. It depends on what you want to know. A negative air machine, you mean you're pulling the air out and, yeah, I mean, I don't know what that does. What I recommend, if you're going to test for spores, you need to go into the space, make as little disturbance as you possibly can, and collect as many samples as you can to get a reliable answer. Now, if you see mold on a wall and you want to know whether it's getting into the air, do what I said before: collect an air sample and then sample the wall to see if it's the same spores. And you can develop a really nice strategy that will tell you whether those spores are getting off the wall and into the air and how far they're going. You can develop a really nice fungal strategy. Whether people will pay for it is another story. But as a research method, I've done it many times, and it's very effective. But running machines, no, I don't recommend doing that. You really, most of the time when you're looking at in a building for mold, you're looking at exposure, and you want to measure exposure. You want to measure the exposure that's really there. Now, if you want to measure that's there all the time, that people are just breathing all the time, if you want to know whether when somebody is in their office and they have a moldy wall, for example, or moldy something, and they bang up against the wall with their chair, if you want to measure that, then you do that. You measure before and then hit the wall with the chair and measure again. So it's a matter of critical thinking, of thinking about what question are you trying to answer. Specifically, very specifically, what question are you trying to answer with sampling? And how is the best way, what is the best way to collect a sample that will answer that specific question? That's what you need to think about. It doesn't come naturally to anybody, really. You have to stop and think about it, and you have to train yourself to think absolutely critically and not just go in and blindly do something somebody told you to do. So that's my recommendation. That's what I talk about all the time, and that's and the fungal strategies seminar really talks about that a lot.
Great. Any final comments before we sign off here, Dr. Birch?
Thank you all, and I apologize for having to go so fast today, but actually I like it better this way because then we can get to questions at the end, and I think the questions are very valuable. Please feel free to send questions, even if I've already answered them. Send them along to David, and I will, if I need to look into it a bit more, I'd be happy to do that. I will try to get your questions answered by the end of next week, because this week is Easter week and it's busy, busy, busy. Okay? Thank you all.
Great. Great. Thanks so much, Dr. Burge. And as a final reminder to our audience members, a certificate of attendance will be provided to those who complete the post-event survey. And the survey will pop open here in just a minute as we end the session. You'll also get a thank you email with that same survey link. If you complete it at the end of this session, no need to complete it again when you get the email. It's the same survey. We're just sending it out at 2 different times. But it is required for you to complete the survey within 24 hours of the conclusion of this webinar. So please make sure you complete the survey as soon as possible. Again, questions about certificates of attendance can be emailed to webinars@emlabpk.com. And again, content-related questions can go to Dave Gallup at dgallup@emlabpk.com. This concludes today's webinar. Thank you for joining us, and you may now all disconnect. Have a great day.